Hello, everybody, and welcome to today's ARI Seminar. My name's Fern Hames. I'm the director at ARI, and I'm absolutely delighted to welcome you all to today's seminar. Pretty excited about this one. First of all, though, of course, I would like to acknowledge I'm joining you from the lands of the Wurundjeri people. It's a pretty grey old day on Wurundjeri country, but it's fabulous country, of course, and I'd like to pay my respects to traditional owners, past and present, to any emerging leaders, to all of the different country on which people are joining us from today. I know there's a wide range of people scattered across the place. And also pay my respects and acknowledge any First Nations people who are with us today. It feels a bit like cultural seasons are shifting, where we've been in the dry season with grasses flowering and butterflies everywhere. Everywhere I walk, there's just clouds of butterflies everywhere, but it really feels like the season is shifting a bit now, and we might be going into eel season, which is pretty exciting too. So to launch us into today's seminar, we've got two speakers. We've got Nick Porch from Deakin, and we've got Peter Menkhorst from here at ARI. One of the things that I think is fascinating that links these two perhaps apparently disparate seminars today is that they're both about things that are not really normally evidently right in front of us. So whether that's visually, spatially, or temporally, these are things that are a bit outside of our more immediate world, if you like. They're things that we have to make a bit more effort to understand and know about. We have to look more closely. We have to examine the past more closely. We have to examine different things with a bit more attention to understand these other things that are part of our knowledge and part of our world. So I'm really excited to hear both these speakers. Thanks to both of you. As Andy said, if you have questions through the way, post them in the chat. We'll get to the ones that we can. And we'll switch over in the middle from Nick to Peter, and then we'll deal with questions at the end. But I'm going to mute now and pass over to Nick. Thanks so much for joining us, Nick. Can't wait to watch your presentation. Go for it.
Thanks so much for that introduction. These things tend not to be so well known, as Fern nicely introduced, I think, and we tend to think about invertebrates often in terms of the charismatic sort of taxa that we might have in the world around us. So Fern mentioned the butterflies that are so common at the moment, but I'm really interested in a lot of the small things, the things that might go less noticed. So something like this dragon springtail, for example, although it's incredibly beautiful, it's colourful and distinctive, probably not many of us, if any of us, have actually seen one before. And they're quite, I won't say they're common, but they're moderately abundant in wet forests in eastern Australia. And on mainland Australia, where they range from the Otway Ranges through Macedon and those sorts of places, the Central Highlands, all the way up to Fraser Island, effectively. They're diverse, but they're almost, well, I won't say they're almost all undescribed, they are all undescribed. So in some ways, and this is reflecting on something that Fern mentioned kind of nicely in the introduction, is they're out of sight, but they're also out of mind. And that's something that I think we need to change if we're really interested in thinking about this idea of using terrestrial invertebrates for doing conservation-based research, or even doing conservation and trying to protect our biodiversity in its biggest picture sense into the future. I've been interested in invertebrates for a long time, and I'm trying to think about how I really got interested in short-range endemic invertebrates, and we'll talk about that in a moment in terms of what I mean there. But much of my research, before I was distracted by bushfire projects, for example, on Australian invertebrates, was really about looking at the nature of invertebrate faunas in fossil records on oceanic islands. And this picture here is me sitting on the summit of one of the mountains on Rylah, which is a little island to the south of Tahiti. It's about 10 kilometres across. And when we look at the fossil record, so the historical evidence of the biodiversity of a place like Rylah, almost all the species that were present there are gone. So the endemic insects, largely gone. The endemic land snails are largely gone. And the only place we can find them is in the fossil record. So something's happened in this system for these species to be so heavily impacted that they disappear from the landscape. And the reason why I'm talking about this is because these systems are relatively small and spatially constrained. That is much like the systems I'm going to talk about when we think about small distribution invertebrates. So even though the drivers of these extinction events on these islands might be different, the fact that these small systems are going to be impacted in Australia into the future by changed fire regimes, by climate change and all sorts of other things, by invasive taxa, then we sort of may think that the threats that exist to these systems are going to be somewhat similar. We also need to think about what we know. And I think this paper that came out in 2017 was a really interesting one, and I really like the way in which they plotted the evidence that they were dealing with. And they were thinking here about how much knowledge is in each of these major groups of organisms. And the taxa in the green at the top, on the right-hand side of the diagram, are ones that have many more records than you would expect based upon their diversity. So a group like birds, which are actually not very diverse, only 10,000 species. It sounds weird to say that, but they're not that diverse. And they're incredibly charismatic and people love them. They record them all the time in terms of distribution. They're incredibly overrepresented compared to what you might imagine they should be based upon that diversity. So in the green at the top, everything there is vertebrates and seed plants, except for one group, which is the Globothalamia, which are foraminifera. And the reason why they are overrepresented is because they're very important in terms of marine ecology, particularly looking at the history of the oceans, for example. So they're well studied. At the bottom end, to the bottom left, we can see that there's three groups in particular that I've highlighted: the gastropods, the snails, arachnids, spiders, mites, scorpions, etc., and the insects in particular are the least well-sampled groups. I don't think there's any surprises there, right? I think that's not surprising at all. So why would we consider groups like this that are really fairly undersampled and we don't know a lot about much of the diversity? Well, there's some really good reasons. One, obviously, is they're really important in food chains and ecosystem function. They have an intrinsic value that I think is important to recognize. And the one that I'm going to focus on is this one that's got to do with the scale at which they use landscapes. Terrestrial invertebrates use landscapes in a different way to most of the other taxa that we might be interested in, which often have much wider ranges. So something like this delid mite here, which is feeding on a springtail, this sort of predation event is happening in a square metre hundreds of times every day. It's fairly remarkable. Those little yellow triangles you can see down here at the bottom, they're actually grains of pollen or spores. They're probably Cyathea or Dixonia spores. So these sorts of, the scale at which this is occurring is very different. And what that means is that these taxa can use a landscape at a different scale, on a much finer scale. So what do we mean by short range endemic? Well, really, a fairly important paper that was published a number of decades ago now defined short range endemics. It was published by Mark Harvey, who specializes in groups like pseudoscorpions and millipedes and the like. He said that they're taxa that have a range of less than 10,000 kilometers squared. And that doesn't sound tiny at all, but really that's sort of not much bigger than the size of Melbourne. 100 by 100 kilometers equals 10,000. So you're dealing with something relatively small in that respect. And this is really common in invertebrates. It's much less common in vertebrates. It's present in lots of seed plants, for example, but it's really common in invertebrates because many taxa are flightless and many are very poor dispersers. And when you're tiny, it's hard to get around. So this thing here, this is an allothyrid mite. Allothyrid mites are one of the larger mites. It's probably up to 4 or 5 millimeters long, which is quite big. And Krantz and Walter, in their book on acarology, say that numerous undescribed species and generic level taxa occur in Australia in this particular group. And that's true. And that's true not only of that group, but it's true across the spectrum. Some of you might recognise that thing in the top left. It's a group that there's a bit of work on at ARI. But the rest of these things here, which make up most of animal biodiversity, if you think about the groups that they belong to, they're incredibly diverse, and they include a large number of short-range endemic species. So something like the Gippsland black snail, Victophanta actromentari on the top right, relatively small range. Similarly, many of these other organisms here all have short ranges. We don't know about their diversity particularly well, necessarily. And this is something that is relatively easy to fix in some ways if we took the right approach. So I guess the next question then is, well, we probably don't need to care about them if they're not vulnerable to the sorts of changes that are happening in ecosystems. And I thought of a really interesting example that got me thinking about this was when we were doing a couple of bushfire projects. We were sampling in the Alps, for example, over 2019-20. And one of the groups that we collected lots of specimens of in a few sites were these really large charismatic pill millipedes. They require deep forest litter, and they're very fire intolerant. If you have lots of fire, you get rid of the deep litter that they require, so it's not surprising. And they have a really weird distribution in Victoria. If you have a look at these records that are pulled together from citizen science, like iNaturalist from the Museum of Victoria, 14 records from each of those, we got 351 specimens in our bushfire recovery project. And if you have a look at the response to fire on the left-hand side, unburnt is the left column and burnt is the right column. They do not like being burnt. And if you burn this potential burnt site again in a few years, you'll likely knock out the last few remaining specimens. This distribution that we see here, it's actually really weird. That's not a normal distribution for a group of invertebrates or almost anything. There's little patches in the Grampians, the southern Gippsland Highlands, the Prom, and some of the eastern highlands. And that's probably reflecting the fact that this group is relictual. It's lost much of the diversity that was present across this landscape in historical or probably going back tens of thousands, hundreds of thousands, millions of years. And what we've got left is the taxa that have managed to survive that long-term drying that happened across the Australian landscape. The reality is we don't actually know how many species are in this group. There's one described, but are the ones from the Grampians different species from the ones in the Prom and South Gippsland and the Eastern Highlands? We don't know. And from a conservation perspective, presumably that's important to know. This sort of pattern is common across the spectrum in invertebrates, which is probably nothing that surprises any of us here. What we can start to do is we can look at published records from recently revised groups, and that's what I've done here. I've looked at the groups where I have revisions that have been published in the last, in this case, in the last 15 years, and I've just chosen four groups in this case. So they're all beetles. I'm a beetle person, or I'm more knowledgeable about beetles than any other group. And I've looked at where the species occur, and particularly where the narrow range endemic species occur. And in these four groups, pretty much everything is narrow range endemic. So we have over in the Grampians, for example, we have 3 species of Tympanagaster. Tympanagaster live on rocky waterfalls and the like, in the algae that live along the sides of the streams. We have Annelina, which are little blind ground beetles that live in the soil and the leaf litter on the surface of the soil. We have Paraptictus, which is a beetle which lives in leaf litter, and Nargiotes, which is another predatory beetle that lives in leaf litter. And if you have a look at the patterns, you can start to see there's patterns that are repeated in each of these groups. And what I've done is I've brought them all together to say, which places have multiple species of narrow range endemic or short range endemic species in a particular place. So somewhere like the Central Highlands, the Upland, the Alps, the Aranundra, they all have species in all 4 of these groups that are only found in those places. The narrower the lines that you see around the particular groups, the fewer present. So what that's telling us all of a sudden is that we have a really geographical structure that we can use to try and think about, well, what places are important for biodiversity in these groups. So what I'm going to do very quickly is focus on 3 areas: the Otways, East Gippsland in southern New South Wales, and then focus in on the Central Highlands, just to look at the way in which narrow range endemic or short range endemic biodiversity is structured in these groups. 2019, pre-COVID, pre-bushfires, I had an idea that it would be really nice to know about baseline biodiversity and leaf litter. And I obviously chose the wrong place because Otways didn't burn. I should have chosen somewhere further east. But that doesn't matter, I guess, because the question here is, how can we use leaf litter sampling to understand the distribution of, I was particularly interested in the short range endemic species in this region. This is color coded by habitat, so wet forests are in green, drier forests are shown in the red, and up at the right hand end we have the Otways Heath, which is at the driest end of the spectrum. And from these 97 samples, which were 2 meters squared of leaf litter, there was 20,000 beetles, 550 species, and at least 15 described short range endemic species. So what you can do once you start to get some more data for these things, you can start to plot their distribution and look at and develop models. This is a Maxent model, for example, for the distribution of this particular species, Dichinotus lymbatus, which is only found in the Otways. And here's another one, Theorella newtoni, which is again only found in this part of the world. But what this particular data set showed was that in a topographically simple range, I don't mean the species range, I mean the topography of the Otways range, you get a simple pattern. The species are widely distributed across the range. What happens when you go somewhere else, when you've got topographic complexity? And this comes from our bushfire work, 2 projects, one led by Don Driscoll here at Deakin and one led by Eloise Gibb at La Trobe, and I've brought the data for one group together. And this is a genus, or a genus in the broad sense of a little tiny cryptic cryptorhynchine weevils. They're about 2 mils long or slightly less. And very clearly, from a morphological point of view, there's different species all the way up the coast, from Erynundra to the Bigger Valley, all the way up, heading across the samples we looked at. Nanyong range has different species to the Erynundra, for example. Mount Dedic likely has a separate species. So we start to see this pattern of really strong topographic drivers of the species that we see in the landscape. If we hone in more closely to where we are at the moment, then Central Highlands and South Gippsland, we have a genus called Tropidotreccus, which has 4 described species. 2 up at Donabuang, 1 in the Dandenongs, 1 called Borborensis in the Borbor. But you start looking in between and in places where people have never looked, and you start finding lots more species in this genus. So in an area where, if we were thinking about vertebrates or plants, we'd think Central Highlands as an entity, invertebrates are dividing that landscape in a much finer scale. To the extent that we find species like the one down here in South Gippsland, it's in a very divided landscape, such that there's very little of the habitat that it requires remaining. This is a really interesting group because Tropidotreccus itself, you might be able to see in this diagram in the bottom, this is a phylogeny that was published with colleagues a few years ago now. It's sister to a group across the planet which has 2,500 species and divides very clearly into a southern hemisphere group and a northern hemisphere group. It means it's a really ancient lineage, and from a conservation point of view, this phylogenetic age is probably something that we should be thinking about. Here's a tweet from a colleague of mine at Harvard who works on harvestmen. Here are 3 species of the Australian genus Trainobunus. All of them are new. In a genus like this, every mountain, every place you stop and collect has a different species. So there's a pattern forming, right? This pattern of short-range endemism at high spatial resolution. So how do we come to grips with this diversity? Well, the fauna is massive, but I think by studying well-chosen taxa and strategically sampling landscape, we can rapidly understand which places are important, and this would involve both morphological and molecular approaches. The project that I was involved with the Latrobe crew, Eloise Gibb and Nick Murphy, for example, were using these molecular approaches to look at difficult taxa. And in the context of future ecosystem change, we can then examine which places or faunas are at risk. And the reality, of course, is, if we think about this logically, the places that have elevated short-range endemism in terrestrial invertebrates, they're demonstrating stability in the long term, are likely to be climate refuges for all taxa. The vertebrates, the plants, and the other taxa that we might be interested in, for example. So just to finish up, if evidence-based conservation of biodiversity is our goal, and I think probably most of us would say yes, that's still true, evidence of large but little studied groups is critical. I think a lot of people think this is too hard, but this is not too hard because I think we can deal with this in meaningful ways. The structure of the landscape and climate history have determined the patterns of elevated short-range endemism. We just need to make the process of understanding those patterns our job. And this probably doesn't make our job in terms of biodiversity conservation broadly easier, but it's certainly evidence-based and it surely makes that more meaningful, I think, into the long term. So just a few acknowledgements. Thank you for the people I mentioned, the funding from the federal government, and a few people for helping out with identifying things. And just to finish up, I'm going to leave you from a quote from someone who we might know. And I think it's relevant both to what I talked about and to everything that goes beyond that. So thank you very much. Any questions?
Thank you so much, Nick. That is just awesome. Like, I love this sense of, it's almost like when you go snorkeling for the first time and you discover this other whole world that you haven't really known is there until you actually look. And of course, your gorgeous photos have really helped give great visibility to this largely invisible world for us. So this is awesome. And this message about those refugia and places where these things persist is an obvious one to us, really. As soon as you started putting up those maps and things, I was thinking, oh, I reckon the Bobo Plateau is going to be interesting here. So
yeah. The Bobo Plateau is really interesting.
I'll bet. Yeah. Look, let's have a quick, Andy, or Tess, do we have any questions? They're probably only just starting to come in because people are probably going, oh, mind blown, before they think about questions. But here we go. Andy, do you want to lead the questions, or is Tess going to do this? We've got a question from Jerry.
Can do. We're expecting an inundation of questions, so I've got Tess filtering them out, but they are just starting to trickle through. The first question is from Jerry Alexander, and he's asking how many people in natural environment programs consider invertebrates as part of the natural resource management. Do you work closely with natural resource managers, Nick, in advising sort of invertebrate surveys and things like that?
I haven't been. I've sort of switched over to this whole business of working on Australian material relatively recently, largely driven, I think, by A, the recognition that there's not much work being done in this space, and B, by the bushfire projects. And I've only literally just finished sorting beetles from the bushfire projects. I think we sorted 70,000 specimens. I think we had about 1,500 species, maybe 1,700 species. But one of the things we need to do, and this is why I'm interested in talking to organisations like yours, is to do more of this work in that space, I think.
Awesome. Still waiting for a few more questions, so I'm going to ask one for myself. You mentioned in one of the research studies that you did that you used a bit of Australian Museum data, but also stuff from iNaturalist to complement, I guess, your bushfire surveys. How important are community observations for your work and learning about these different patterns happening across lots of different spatial scales?
I think for some groups they're very important. So groups like pillimillipedes, which are probably instantly recognisable, they're relatively large, for example. For groups like that, they're important because people will post things like that online and we can identify them relatively easily. For other charismatic groups that are large, that people can identify, things like bogling moths, for example, things like Christmas beetles that have been in the news a lot over the last few years, they're recovering really well as it's wetted up rather than being dry. But for most of the diversity that I'm interested in, so let's say of those 1,700 beetles we identified during the bushfire recovery projects, very few of them would be identifiable from a picture taken by somebody in the field, for example. There's a lot more magnification, for example, that might be required. So that's partly where it's difficult. There's an overlap at the large end of the spectrum, but as you get down to the really rich diversity, it gets much more tricky.
So it only helps at a sort of a really broad scale.
Yeah, but still, I mean, I think I had a picture somewhere in one of my previous slides, maybe that one actually, that beetle at the top there. That's an undescribed species and perhaps genus that's known from the Alps of Victoria. And I remember seeing it many, many years ago at Dinner Plain, and I was up at Dinner Plain during the bushfire recovery projects and saw lots of them. And it's one of the things that's on iNaturalist. People have taken photos of it going, what's this big, beautiful beetle? But they can't identify it because it doesn't have a name. So that sort of limitation also is very difficult. They can put a group name on it, like a family name, but beyond that, it can be quite tricky.
That's a really good example. Thank you for that one. I've got a question from Lindy. How much do you think that some of these big gaps in distributions are due to not enough people looking, or if they're real gaps in distribution, especially when you're sort of clarifying that these things are hard to identify?
Yeah, that's a really good question. This is something that basically inspired thinking about the impacts of the bushfires on these groups. And when Don and I were putting together one of the proposals, basically I was thinking, are we overselling the likelihood of narrow range endemism in these groups across the landscape at the scale that we're sampling? In retrospect, I think we undersold the scale and that we should have sampled on an even finer scale. So in places there'll be big gaps, and I think what I'm talking about mostly in terms of the examples that I've given is wet forest taxa. So wet forest taxa divide the landscape up in a very different way, largely because wet forests were characteristic of the Pliocene and beyond, and they've been contracting into little habitat patches. And that's why I think when I mentioned my example about Rayvarvi, we have lots of little islands of habitat, of wet forest habitat. And at what scale do those islands still contain endemic species? We're not really sure in many ways. So I think we're still at that we're certainly still at that discovery stage in many groups of trying to understand what's going on.
Awesome. Thanks, Nick. I've got to jump to Madeline's question because you mentioned how fire has a large impact on a lot of species that aren't tolerant to fire. Going forward, how do we sort of work hand in hand with invertebrates' needs and the role of ecological fire and cultural fire burns in the landscape?
Well, I think this is one of the things that the reason I chose the pillimillipedes was because it's a group that's, although it's not well known where we are in Victoria, it's well known in southwestern Australia, where there's two species that are both relict. They're both restricted to the wettest forest patches. And what the sort of management recommendations that have been made, there have been to do relatively low impact burning during seasons when you're not going to basically fry everything. And this is one of the things that I think people talk about a lot in terms of Indigenous fire management and the like going forward, is that it's a way which we can perhaps better manage these taxa into the future. The fact that they're with us today means that that whole process has worked for at least 50 to 60,000 years. It's worked really well. So it must be able to work really well into the future, I would think.
Really good point. And I really like how that intersects with that climate refuge idea you had, and trying to avoid repeat burns when things are vulnerable. I've got one last question I think we have time for. This one is about, it's particularly in East Gippsland. It's actually really similar. It's about reinitiating fuel reduction across East Gippsland without compromising invertebrate biodiversity. I feel like you've answered that kind of already with your description. Is there any other sort of aspect in terms of managed fire or responding to big bushfires that you've been working on that you'd like to add?
Yeah, I guess one of the problems with thinking about fire is that even with taxa that are really, really well known, like the vertebrates, the plants and the like, we still probably in many cases don't really understand what the requirements are necessarily. If you've got a group of organisms that are really poorly known, not well studied, it's even harder. We did find different patterns in terms of the response of short-range endemic species along the escarpment, so sites like Errinundra and then along the wet forest, the rainforest of the escarpment, versus the high mountains. The high mountain taxa seem to cope better with fire than the ones along the escarpment, and that makes sense, I think. Much more open habitats, which probably have more fire in them more frequently, whereas the escarpment sites, being wetter sites, being closer to the coast, and being often summer rainfall sites, don't have that fire. So there may be patterns like that that exist in the data, I think, and that's something we're working on at the moment.
Thank you very much for those answers. And I'm going to throw it to Fern to ask the final question, because I can see hers has popped up in the chat too.
Yeah, I couldn't help thinking about this one, Andy, because like what you've just done, Nick, is completely opened our eyes and awareness for those who weren't already understanding of this, of all of these issues and how important this is. And I think any of us who've ever listened to a whole lot of other advocates for inverts totally would be on board with you. But we, as Jerry and others' questions have indicated, we're still struggling to get these on the table in terms of people caring about them and investing in them and doing the work that we need to do. So what do we do? How do we nudge these further up the list of being considered for research investment, for protection, for all of the things that we need to do? How do we increase their visibility?
Well, this is one of the reasons why I'm here, I think.
Yes, I should say that I started my question by saying, apart from fabulous presentations like yours, what are the things that we need to do? It
is a very good question. I think it's a really difficult one, and it's something that I think is becoming driven by other people. Zoos Victoria, for example, has now got an invertebrate conservation biologist position, largely driven, I think, by advocacy coming from people within the organisation saying that we're not doing anything about these groups that are being heavily impacted by fires, for example. And I think a lot of that was driven by the bushfire, the bushfires of 2019, 2020. Beyond that, yeah, I really don't know how we go about changing things in this sense, because it is a stepwise change in some ways. Just the number of things we're talking about. I mean, terrestrial invertebrates are more than 95% of animals in the habitats that we have in Victoria and beyond. So it's really difficult, and I think even within an institution like mine, I teach into the wildlife and conservation biology degree. There is not a single unit, for example, that teaches students about the diversity and management of organisms like this. So I find it remarkable, but that's just the way the world is.
That is remarkable. And now everybody on this call today has a responsibility, right? We all have a responsibility to do something about this and advocate for little critters in the leaf litter.
Absolutely, I agree.
All right, we've got a few champions on board and we're going to keep growing it, Nick. I'm going to close it out there. Thanks again. Thank you so much for an awesome presentation. That was wonderful. I suspect that questions will probably continue to pop up in the chat as people process what you've been saying. But we're now going to switch over to Peter, who's going to give us a slightly different lens on things that we're perhaps not quite aware of in the way that we connect with knowledge and the world. Peter, are you with us? And yes, Andy's saying 154 new advocates. Yes. And we'll double that when people start watching the recording as well. Peter, here he is. Hi, Peter. Thanks so much for joining us today. I'm assuming that many of you will know Peter Menkhorst from ARI. He hates it when I call him a legend, but he's an absolute legend of our staff at ARI, and really privileged to have the opportunity to listen to him share some of his wisdom today. So over to you, Peter. Thank you.
Thanks, Fern. And I'll begin with a welcome to country, an acknowledgement of country, sorry. I'm speaking from the lands of the Wurundjeri people, and I wish to acknowledge them as traditional owners and pay my respects to their elders, past and present, and to the Aboriginal elders of other communities who may have joined us today. I also want to point out that I'm we'll be talking about Surveyor General Thomas Mitchell. And I recognise that he is a challenging person for some people, particularly traditional owner communities. But the collection of an interesting mammal specimen by members of his party is central to the story today. So in this presentation, I'll begin with a brief introduction to ecological history, and then a brief reminder about the rate of loss of small to medium-sized mammals in Australia. And thirdly, I'll discuss a local example of how detailed examination of historical records can lead to revised conservation strategies for species. And then I'll draw some broader lessons from that work. And so ecological history is really just interpreting historical records through an ecological lens. And the sources of ecological information are very diverse and can be almost anything. And often they're from sources that were documented for an entirely different purpose, such as personal reminiscences. And this means that it's really important to take care with interpretation of the narrative. There's a real need to avoid over-interpretation of what may have been incidental comments or unintentional silences on a particular subject. And despite these caveats, information gleaned from diverse historical sources and interpreted from an ecological perspective has the potential to shed light on the pre-European condition, if you like, such as the distribution and habitat of species that have declined since European arrival, and even the drivers of those declines. And a lot of people have done this sort of work, notably Ian Abbott in Western Australia, and in Victoria, particularly Rowan Bilney in Gippsland. And he used mainly subfossil materials from caves and owl deposits and so on. Whereas I'm going to be talking about an example from northwestern Victoria, much drier country and not the sort of topography that provides dry refuges for bone material to survive for long periods. And down on the slide there, you'll see an example of ecological history in an interpretation that I did in 2008, 2009 of Gerard Kreff's wonderful paper on the vertebrated animals of the lower Murray and Darling rivers, which I'll talk about a little later. So just the Australian mammal decline, we're all very aware that Australia holds the record amongst all nations for recent mammal extinctions. Victoria's contributed its share to that record. About 18% of Victoria's 133 non-marine mammals are no longer present in the state, and most of the remaining taxa have greatly reduced distributions. The graph on the right there is from John Winarski's work, just plotting the cumulative total of mammal extinctions through time. The open circles, the lower line, is at the species level, and the upper line has subspecies added to the species total. And you can see that extinctions began very early in European settlement. Even by the 1840s, things were disappearing. And that's well before the arrival of introduced herbivores, introduced predators, rabbits, and so on. But after the introduction of large numbers of livestock across an incredibly, surprisingly large part of Victoria, and also after the disruption of traditional owner land management practices. But it's not my purpose today to talk about the causes of those declines. Rather, I want to talk about the need to recognise how early some of them happened and what implications that there have been. In particular, we need to recognise that most of these declines went unnoticed and undocumented. And this means even right up into the 20th century, almost to the mid-20th century, things were declining and those declines were not being recognised. So this means that the current distributions may not represent the original distribution. Currently occupied habitat may be a subset of the original, to the extent that it's now difficult to define the natural mammal assemblage in a given area. And the species that have survived likely did so because, well, they survived in areas where the combination of threats that were acting on them had the least impact. And these kind of considerations are really relevant to programs aimed at establishing functioning mammal communities. So the example that I'm going to use relates to Mitchell's hopping mouse, a member of the genus Notomes. There are 10 described species, half of them are extinct. Mitchell's hopping mouse weighs between 10 and 50 grams. It's quite a large animal for a hopping mouse. An important part of their life history is they live communally in deep burrows, and they eat a range of plant material and possibly also insects. So Mitchell's hopping mouse has this southerly distribution across Australia. I'm going to be talking about this easternmost area of the distribution in the Murray Mallee of Victoria and South Australia. And here we think of them as inhabiting deep sands, the jumbled dune systems of the Big Desert in parts of the Sunset Country, at a Colquhoun National Park and Nuello Flora and Fauna Reserve, for example. So just remember that photo of deep sand dunes with that wind-blown siliceous soil. Now, Mitchell's hopping mouse has sort of perplexed me for about 40 years or more since I realised that the type specimen was collected during Major Mitchell's 1836 expedition through what is now Western New South Wales and Western Victoria. In fact, the type specimen comes from near Lake Boga, south of Swan Hill, indicated by the red arrow. And that locality is 115 kilometres southeast of the nearest modern known population in Anuello Flora and Fauna Reserve. And it's in a different bioregion to where the vast majority of modern records come from. So the modern records are mostly in the Lowen Mallee bioregion. That's the orangey colours here. These green dots are records from the VBA in recent decades. But the type specimen is way out on the eastern edge of the Murray Mallee, perhaps even in the Murray Fans bioregion, and even close to the Riverina bioregion. So there's something of an anomaly there. And in order to try and understand that, I've been looking at the historical records of two 19th century exploratory surveys. And they are Major Mitchell's expedition, as I mentioned, and also the Blandowski expedition that happened 20 years after Major Mitchell went through. The sources of information that I've used are Mitchell's book, titled 3 Expeditions into the Interior of New South Wales, published in 1839. And more recently, as part of the celebrations of the 150th anniversary of Mitchell's expedition, a surveyor called Gregory Eccleston was employed by the Victorian government to do a detailed mapping of Mitchell's route across Victoria. And he produced a report that includes these wonderful maps showing the exact distribution of Mitchell's party, the exact route that Mitchell's party took, and the location of their overnight camps and so on. And more recently, Eccleston was able to also locate in England the diary of Mitchell's second in charge, Stapleton, and he then interpreted Stapleton's diaries, and that provided further information about the capture of the hopping mouse and other interesting specimens that Mitchell's party collected. And this illustration in the bottom left there is Mitchell's drawing of Notomis mitchellii, Mitchell's hopping mouse, and that illustration was used as the basis for the description of the species by Ogilvie at the British Museum. And then there's the Blandowski expedition, which 20 years later travelled from Melbourne up to the Murray and downstream along the Murray to near its junction with the Darling, where they established a camp and stayed there for 7 months or so. And they saw the sources of information about the Blandowski expedition, Gerard Kreff's wonderful paper from 1862, which I showed you before, on the vertebrated animals of the lower Darling and Murray, the lower Murray and Darling rivers. I think the most interesting local fauna ever written in Victoria. And then again, as part of the 150th anniversary celebrations of the Blandowski expedition, the Royal Society held a conference in 2008, and they published the proceedings of that conference in 2009. So if you want to know more about the Blandowski expedition, volume 121 of the Proceedings of the Royal Society is available open access, I think, online. So I've been dredging through those historical sources of information. I did a lot of this work during lockdown. It's not actually an official ARI project. So getting back to Mitchell's journey and the type specimen, this map shows his route through Australia, through Victoria. He travelled very widely, as you can see, right down to the coast at Nelson and down to Portland, where he bumped into the illegal settlement by Henty and others. And then he returned diagonally across the inland slopes of Victoria, basically, back to Sydney. But I'm really only interested in one day of that big journey, and that's the 21st of June, 1836, when Mitchell's party travelled from Swan Hill, where they had spent the night at the junction of the Murray River and the Little Murray River. Mitchell coined the name Swan Hill at that time, and they travelled down past Lake Boga to where they camped near current-day Fish Point. So here's a more detailed map showing their route. That's the dotted black line, as plotted by Eccleston. Importantly, the pink zone there on that map is my estimation of the type locality of Mitchell's hopping mouse. I believe it was collected somewhere along that path, probably on the morning of the 21st of June. And I don't have time to go into all the reasons for that here. It's not particularly pertinent, but the exact locality of the type specimen has been variously described as anywhere from the junction of the Murray and the Murrumbidgee, which is more than 100 miles, 100 kilometres north of here, to an area down around Tresco to the southeast of Lake Boga. But it makes much more ecological sense that it happened. The capture happened along the boundary of the Murray Fans, the floodplain of the rivers, and the Murray-Mallee bioregion, where there are red sand dunes, which is much more likely to be a hopping mouse habitat than the clay soils of the floodplain. Mostly because the hopping mice burrow, and there's no point in burrowing in areas that are going to be inundated. Now, the Blandowski expedition. William Blandowski is a fascinating character. He was Victoria's first government zoologist, in a similar way to the government botanist, whose name escapes me. Someone can put it in the chat. Another German. So Blandowski led Victoria's first wildlife survey, and it was a really important wildlife survey and was really not matched in its intensity and its outcomes on Muir, thank you, for about 120 years till the Land Conservation Council was formed and people at ARI and the Museum of Victoria and elsewhere started doing detailed fauna surveys across the state. So between April and November 1857, the Blandowski expedition was based at Mondamilum, which is in the current township of Merbein. Here's a picture of their encampment. And Blandowski was quite clever in that he paid the local Aboriginal people to collect fauna specimens for him. And here's a rather fanciful illustration that was drawn many years later in Europe at Blandowski's request, showing him in sort of heroic pose here, receiving specimens that have been brought in by the local Nyeri people and others from further north along the Darling. And I presume this character here, the character in the bottom left is Jared Kreft receiving the specimens. Now, the important thing about the Blandowski results for this little story is that they collected 96 specimens. Well, 96 specimens were catalogued by Kreft, and Kreft wrote in his paper that the hopping mice were very plentiful on the Darling, and as many as 50 specimens were often procured by the native women in an afternoon. It burrows into the ground and is dug out by them. Amongst the specimens catalogued by Kreft, there are some notable absences, particularly the Lowen Mallee specialists, the silky mouse, the little pygmy possum, and the Mallee Ningawi. So I take that to mean that the Lowen Mallee bioregion, remember back to that map of the bioregions, that was the orangey brown area where the hopping mice specimens are concentrated, was not sampled during the Blandowski expedition. So therefore those hopping mouse specimens must have come from outside the currently occupied habitat. And they likely came from big mallee eucalypt woodlands growing on the sandy loams of the Murray-Mallee subregion, and maybe from colitrus woodlands and even chenopod shrublands of the Murray Fans and Pooncarry, Darling and Menindee subregions in New South Wales. So that says to me that we need to broaden our thinking about what is the habitat of Mitchell's hopping mouse. From this photo that I showed you earlier of the deep sand dunes of the Big Desert to include tall mallee areas in the Murray-Mallee bioregion, which has been massively cleared for agriculture, of course. It's now mostly wheat farms. And even to include areas on the Darling, as Kreft described them, which include saltbush, shrubland and colitrus woodlands. So the conservation implications of these sorts of considerations are that the habitat of Mitchell's hopping mouse in southeastern Australia extended far beyond that currently occupied, to include at least 6 other biogeographic subregions. So planning for its conservation should now include a greater breadth of habitat and a huge increase in the area of its former distribution. And this has been recognised by the New South Wales Government and the Australian Wildlife Conservancy in their program at Mallee Cliffs National Park, which is northeast of Mildura, where they are actually currently introducing Mitchell's hopping mouse and other species into a predator exclusion area, a very large predator exclusion area. And the hopping mice that they're using for that translocation were the progeny of animals that they collected from the Big Desert in Victoria. So conclusions from this work are that the Mitchell's hopping mouse case study that I've just described emphasises the value of considering ecological history. I'm sure that other species would benefit from similar attention to historical literature. And ecological history often brings critical insights. It may reveal and counteract the incremental loss of knowledge over time. For example, the trap of tempocentrism, which is the loss of community memory and the consequential reduced understanding of the original condition. And I would argue that time spent researching ecological history of species and areas will almost certainly provide valuable insights for conservation biologists and conservation planning. So we need to cast a wide net and capture all available sources of information. And I'll leave it there with some acknowledgements to Gregory Eccleston for his scholarship, deciphering Major Mitchell and Stapleton's notes and plotting the course taken, to Harry Allen from the University of Auckland for driving the 2008 Landowski Expedition Symposium, and to colleagues at ARI for helping prepare the maps, and to Andy, Lindy and Fern for helping sort out my jumbled messages and thoughts. Thanks for listening.
Thanks, Peter. I just find this so inspiring, this reminder that, you know, how we see and perceive the world right now is a bit of a trap, and that we do need to think not only, like Nick suggested, you know, look more closely, but also look back, look in other directions, look at how other people have described and seen the world as well. This is such a really useful reminder and quite inspiring. Thank you so much for all of that. So I imagine it's going to take a minute for, I know, here they come. It might have taken a while for the questions to come through, but there's at least a few comments coming through. Andy, have we got any questions in those lots of lovely thank you messages? There's some questions in there.
I've not seen one just yet, but everyone is expressing their just appreciation for you doing this exploratory work, Peter, and looking into an area that many of us wouldn't have the expertise or understanding to do, and just opening up another bit of our world. I just wanted, you sort of talked about that shifting goalposts over time with that temporal centrism concept. Has doing this reflection work on past histories, has that made you think about other species you've worked on in the past or of your career, and maybe reconsider those assumptions you might have made during this sort of period?
Yes, sure. And I have done this sort of ecological history digging on other species, including the helmeted honeyeater, where the type specimen is way outside the current distribution, and records of the night parrot in Victoria. So yes, whenever we were doing fauna surveys during the days of the wildlife survey team, we did make a point of trying to track down historical records. And the VBA includes, in the days when the VBA started out as the Victorian Mammal Atlas, and I was very keen to capture historical records through that process. So, for example, we went right through the Victorian Naturalist and pulled out records of all mammals right back to the start of the Victorian Naturalist, which was in the 1880s, maybe early 90s, 1890s. Helped in that process by Bob Warneke and John Seebeck. So yeah, it's always been a bit of a focus of mine. But I'm sure that any species of Australian wildlife fauna has interesting historical story to it that can shed new insights into its original distribution, habitat, ecology. Yep.
Thanks very much for that. Lou, so I asked a question about the past ones you might have done. Lou's asking what might be the next species you might do a similar sort of research project into. Do you have something on the list?
Well, I've been looking at two other species closely, but unfortunately they're both extinct, so we can't actually do much about that. That's the pig-footed bandicoot, which is I should mention Major Mitchell, a few days before he collected the type specimen of Mitchell's hopping mouse, he collected the type specimen of the pig-footed bandicoot in Victoria. You know, the fabulous pig-footed bandicoot comes from Victoria, was first found in Victoria. So that's another story that's greatly interested me. And the question coming from Louise, I'd say leadbeater's possum, Lou. We really should be looking at, you know, the sort of habitat that occurred in the Bass Valley that leadbeaters, and I know you have been, leadbeaters occurred in, and the Mount Wills area, historical records of leadbeaters way outside their current distribution. I think they probably have a lot to teach us. And the recent surveys that the ARI team have been doing and successfully finding leadbeaters in habitat other than ash forest, I think are important. We really need to understand that distribution outside, you know, what's normally thought of as leadbeater's possum habitat.
Well, we can all look forward to that one for sure. The question from Georgina is talking about early logging and the story that marsupials might have been transported around with timber mills or with the timber to timber mills. Have you heard about any of that in the historic records or any descriptions of that kind of transportation?
No, I haven't. No.
Okay. Well,
not heard
Georgina,
that at all. That's fascinating. I've heard of, you know, people distributing cats around the countryside in the hope that they eat rabbits, but no, not by timber workers as such.
Well, if Georgina has any evidence, maybe she'll share it with you and start a new line of investigation. Yes, as Fern mentioned in chat, tree frogs in banana boxes. Yep. We don't have any further questions in the chat, Fern, so I'm going to pass back to you. Actually, Danny has got one, and we can't ignore Danny's question, so we're going to go to it very quickly. There's been some attempts to identify geographic origins of animals using trace elements or stable isotope signatures. Do you think that could work with these old mammal specimens?
I don't know, Danny. I don't know enough about stable isotopes and whether they would persist in dry mammal skins, what dry museum specimens or wet museum specimens. Yeah, interesting thought.
Indeed.
Maybe someone else has got more idea about that than me.
That's a really good question. Yeah, Nick's saying it should work fine. I'm thinking all the, you know, odorless examinations that some of our ARI colleagues do for stable isotopes to understand where those animals have been. That's a really interesting question, Danny, and we're going to get Nick onto that now.
Yeah. Teeth or bone, yeah.
Yeah, interesting. All the interesting other questions that get raised when we start to pose a few questions. That's really great. And I was actually in the ARI library this morning, Peter, and noticed all those old shelves of VicNat.
Yes.
And
really important.
There's an absolute treasure trove there. It's really fabulous to have access to those sorts of important documents as well. We shouldn't forget what we can learn from those things. Just because they're not online doesn't mean they don't have really valuable records for us.
Absolutely, yes. I mean, I've been interested in the ARI library for 40 years and I know it fairly well.
Yeah. So,
unlike a lot of my colleagues who never go in there.
Well, it's a treasure trove, and it's one of those things you only know what's in there if you go in there and check. All right. Well, some are available online, but not all of them. Thanks, Graham. I might close it there. We're a little bit over time, but we didn't want to cut things off there. Great questions in the chat. Great speakers today. Thank you so much, both Nick and Peter. They were really thought-provoking and interesting talks that widen our minds a bit into think beyond the things that we normally think about. So thank you so much, both of you, for those. Thanks to Andy and Tess for sorting out all the tech for us today. Thank you to all of you who joined in, especially those who posted comments and questions in the chat. But thanks everyone for coming along. We hope to see you next time at our next ARI seminar. Oh, Andy, I've forgotten the date. What's the next one?
Look, you've got me too, Fern. You put me in spot and I can't remember either. Tess.
It's a surprise. Stand by in your email. We'll send the details out super soon. Thanks so much, everybody. Thanks again, Nick and Peter. Bye. Have a great week, everyone.